We have identified a class of molecules that are significantly associated with the risk of prostate cancer and metastatic disease in Black men.
Hello, I'm Emily Schneider from the American Chemical Society, and welcome to this media briefing from ACS Fall 2023. To explore why prostate cancer disproportionately sickens and kills Black men, researchers are looking to another disorder, diabetes, which alters metabolism. They used this approach in a preliminary clinical trial and today report the identification of 4 metabolism-related biomarkers linked to an increased risk of metastatic prostate cancer in men of West African heritage. This discovery could lead to improved testing and treatments for these patients. We're joined today by Sarah Shuck from City of Hope. Thank you for joining us.
Thank you. Thanks so much for inviting me.
Sarah, what were you trying to find out with this research?
So, our lab has been interested for years in understanding why patients with diabetes have an increased risk of developing secondary diseases and specifically why they're at a higher risk of cancer. So here we wanted to apply this important question to see if molecules that were associated with changes in metabolism that result from incidence of diabetes, as well as poor diet and other environmental factors, could be used to assess the risk of prostate cancer development and metastatic disease in Black versus white men. So we were hoping to identify novel biomarkers of disease and also potential therapeutic targets that we could use for precision medicine approaches.
And why is this such an important thing to know?
This is so important to us because Black men are at a significantly higher risk of both developing and dying from prostate cancer. And we really don't know if the tools that we're using to assess risk and diagnose disease have the same utility in different ethnicities. So we're trying to identify markers that have utility in Black men that might allow for more accurate disease risk assessment and develop therapeutic tools to treat disease in these patients.
And how did you get the idea for this project?
Well, we began by establishing a collaboration with doctors Rick Kittles and Leanne Woods Burnham, who are currently at Morehouse School of Medicine. And they had worked on a clinical trial where they enrolled both Black and white men with and without prostate cancer. And over the years, they had collected hundreds of blood samples and also isolated genomic DNA from these patients as well. They also had extensive clinical data on these patients. So after talking with them, we realized that this might provide a really exciting and impactful model to study the role of diabetes and metabolic changes in the risk of prostate cancer.
And how did you investigate this?
We investigated this work through our collaboration with doctors Rick Kittles and Leanne Woods Burnham. So they had performed a clinical trial where they collected samples from Black and white men with and without prostate cancer over several years. This trial collected blood samples and also isolated genomic DNA from these patients as well. And they also collected extensive clinical data on these patients. So we know lots of different factors that they measured in the clinic. And one of the most impactful parts of this study is that Dr. Kittles has developed a very robust genetic test that allows us to determine the exact percentage of West African ancestry within our population. So using this test, we're able to very accurately put people into groups of either Black or white men. And this is a unique aspect of this project because most studies rely on self-reporting to determine race. From this clinical trial, we investigated the presence of these molecules that are produced from changes in metabolism. And we used a novel analytical method that was developed by myself and Dr. John Termini at City of Hope. And we use this method to quantify the levels of these molecules inside the blood from Black and white men with and without prostate cancer. And I just want to mention that one of the unique aspects of this study is that the molecules we are measuring not only have utility in predicting disease risk, but they might also drive disease as well. So we also measured the expression of genetic and protein markers that might be associated with a higher risk of prostate cancer and are related to the production of these metabolic molecules as well.
And what did you find out?
Well, when we began our study, we had originally hypothesized that Black men with prostate cancer would have higher levels of these circulating molecules compared to white men. But when we analyzed the data, we actually found the opposite was true. So we found that Black men had lower levels of these circulating molecules compared to white men. So what we think this might represent is actually in Black men, there may be accumulation of these molecules inside the tumor that may drive tumor growth. So we were also really excited to find that not only were these molecules associated with cancer risk in Black men, but they were also associated with metastatic prostate cancer. And we looked at dozens of other clinical variables that were collected as part of this clinical trial and found that these molecules were the only things that were significantly associated with metastatic prostate cancer risk in Black men. So we also looked at genetic changes that were occurring between the different groups in our study, and we were able to find that genetic changes that were associated with the ability to get rid of these molecules in the blood were also significantly different in Black versus white men. And we also found that there were changes in proteins that are also important for regulating these molecules. So we really were able to take a multifactorial approach to be able to look at lots of different measures that are associated with diabetes and determine how they might be associated with prostate cancer.
And then I guess what's the implications or significance of these findings?
Well, from this study, we identified a novel class of markers that are significantly associated with the risk of both prostate cancer and metastatic prostate cancer in Black men. And we also found unique genetic and protein markers that are associated with this risk. So what it allows us to do is have the opportunity to develop a multi-component test that might have utility in predicting the risk of disease in men that have diabetes or those that might be at higher risk of prostate cancer.
Could this test see any clinical use?
It's our hope that we would be able to implement this test to determine the risk of developing both prostate cancer and metastatic disease in Black and white men. And what we hope to do is implement a multi-component or multifactorial approach to be able to very accurately determine disease risk. And our hope is to be able to use this not only to predict disease, but also as a way to develop precision medicine approaches. So determining which patients will respond best to certain types of therapies.
And then I guess what would be a big advantage of that system over what's currently used?
Well, we hope that we'll be able to predict disease more accurately and earlier than we currently can, and also identify specific markers that work well in Black men as well as white men.
And what's the next step in this research?
So our next step is really based on the surprising finding that there were lower levels of these molecules in the blood in Black versus white men. So our hypothesis now, as I mentioned, is that there is accumulation of these adducts inside the tumor that might exacerbate tumor growth and drive metastasis. So our next step is to take cell lines, prostate cancer cell lines that are derived from either Black or white men, and put them into mice, and then see if we expose these mice to high glucose or factors that are associated with diabetes, can we drive increased tumor growth, and are these molecules accumulating inside the tumor?
Well, thank you so much, Sarah, for sharing your research with us.
Yes, thank you.
Be sure to check out our other media briefings for ACS Fall 2023, which will be posted throughout the meeting at www.acs.org/ACS Fall 2023 briefings.